Okay everyone, welcome back. I'm going to try to show you how to how to form a peptide bond from two amino acids in this video. And this is essentially the same mechanism as the formation of an amide from a carboxylic acid group, in this case on this amino acid here, alanine, and the amino group, the free amino group of a second amino acid, in this case serine. So that's the R chain of the serine, and this is the R chain of the alanine. This chemistry is the same as the formation of the amides that I showed you in the last video, and it is side chain independent. So this has all to do with the amino group of one amino acid and the carboxylic acid group of a second amino acid. Now I've shown you the form of these with a protonated amino group on the alanine and a deprotonated carboxylate on the serine. Now those are the normal ionization states at pH 7, but this reaction is the reaction between an amine and a carboxylic acid, and it's driven by the nucleophilicity of that nitrogen, the excess electrons there. And so if I protonate that amine like this one, we take away the nucleophilicity of that nitrogen. So this is the active form of this thing. And in water at pH 7, there's not going to be very much of that at all. You'll be able to calculate how much of that, you could be able to calculate how much of that using the Henderson-Hasselbalch equation, for example. But there's very little of this available in a water solution at pH 7, and very little of this protonated carboxylic acid at pH 7. But inside of a protein or inside of a ribosome, you can change the pKa of these groups, and you can actually get a significant proportion, enough to react, of these particular amino acids. And the way you do that is, we're going to show you this later in the course when we do enzyme catalysis, but you can pull that proton off with an acidic side chain of the enzyme and sort of make it react as this nucleophile. So for this video though, I just want to go through the chemistry here. This is essentially the formation of a peptide bond by a condensation reaction between the amino group of one amino acid and the carboxylic acid of another. So we start the reaction, as always, in these reactions that I've been showing you, with a nucleophilic attack on that carbonyl group of the carboxylic acid by the electron-rich amino nitrogen. Now that's going to form a bond there, so we have to open up that double bond on the carbonyl, otherwise we'll have 5-bonded carbon, which is of course not allowed. And so our first intermediate here is going to be nothing much happening to this end of the molecule. We still have the protonated amino group here, and as I said, nothing much happening to the side chains, nothing at all happening to the side chains. So the methyl group of alanine remains the same, but what we do have is a change in that carbonyl. Oxygen that's opened up now has a negative charge, and we still have the OH group of carboxylic acid, but now we've got a new bond right here formed from the free lone pair electron there and the carbonyl carbon of the original alanine. Okay, so that's the new bond formed there, and we still have our serine side chain, which is unchanged, side chain independent here, chemistry, and then we have our carboxylic acid or carboxylate group on the serine residue, and I'm just going to shortcut that with the COO minus. Now if we look at this amino nitrogen, we've got 1, 2, 3, 4 bonds there, so this is going to be positively charged. Okay, and so this should all look very, very familiar to you by now. We know we're going to try to protonate this hydroxyl group on there, and so we're going to move that proton over to the oxygen, okay, and that's going to generate another intermediate form here. So starting with the alanine amino group, again nothing much happening, nothing at all happening with these side chains. So we've still got our carboxylic acid carbonyl oxygen as a negative anion here. And now the new thing that we've done is we've protonated that OH group. Okay, and so we've actually got a positively charged oxygen there. And at this point, we've regenerated a neutral nitrogen, still has that hydrogen from the original, its lone pair, and now we've got the side chains of the serine here unchanged, and carboxylate group of the serine again unchanged. So we've managed to neutralize that nitrogen, and we've now created this very good leaving group. And as always in this type of chemistry, this is exactly the same as the amide formation. We can cause that water to leave. It's a very good leaving group, and when it does, we can collapse one of these non-bonded electrons down in and reform that carbonyl double bonded carbon oxygen system. So our next intermediate here, nothing going on with this end of the molecule. We've still got the alanine side chain unchanged. And we've got now our intact carbonyl, and we've got our imido group unchanged side chain of the serine, and of course we have carboxylate of the serine. I'm just going to draw that protonated form here, so I'm cheating a little bit. I'm just going to protonate that so I can make a point in a little bit. Okay, and we did eliminate water here, so I'm just going to balance this out by adding the water. And so this is a condensation reaction, and what we've actually done here is we've formed this peptide bond. This is called a peptide bond when it's between two amino acids. It's a peptide bond. It's an amide, actually an imide, that's a secondary amine that reacted with this carboxylic acid. And when it's between these two amino acids, we call it a peptide bond. Now, you recall last time when I talked about amide formation, we had these equivalent structures called tautomers, and I wrote a few of those out to demonstrate the double bond character between this carbonyl carbon and that imido nitrogen. And we can move that lone pair into a double bond here and move one of these pairs of electrons out to the carboxylic acid carbonyl oxygen, and we get a form that looks like this. So again, I'm talking now about the planar, non-rotatable nature of this peptide bond and how this particular structure here between what was the carbonyl carbon and the imido nitrogen, how these particular bonds, this one right here, has double bond character, and therefore makes it very difficult to have any rotation between those two molecules. And so here, just finishing out the molecule here. Oops, that doesn't look very good. Still doesn't look very good. Double bond carbonyl carboxylate here. Okay, and so this double bond, now we have missing here a hydrogen. So this is a positively charged nitrogen. It's got 1, 2, 3, 4 bonds. And of course we can move that proton over here and make that a COH group. Just look back at the amide videos, same structure there. But this is essentially that double bond nature of that carbonyl carbon and nitrogen of the amido bond here. And so that makes that bond, which is equivalent to that structure right there, not rotatable. This bond here can rotate. We can spin these groups around there, and this bond here can rotate. Those are fully rotatable. But this peptide bond, it remains flat, planar, and not rotatable. Okay? And so this is one of the very great characteristics of a peptide bond that you should be familiar with. This is flat, planar, and there's very little, if any, rotation of this bond right here because of that double bond character. Now, one thing about peptide bonds, like all the other bonds I've been talking about, all these reactions are reversible. We can actually take this water and we can nucleophilically attack that carbonyl carbon right there. We can regenerate this positively charged oxygen here. We can move the proton over to the amino group, like here, and then we can eliminate one amino group from that carboxylic acid and regenerate our serine and our alanine from this dipeptide. This reverse reaction is the reaction that proteases catalyze. They actually add that water back into the peptide bond, hydrolyze it, so a protease is a peptide hydrolase enzyme. And of course in the forward reaction, that's the formation of a peptide bond. This is the chemistry behind how you form polypeptides. Okay, and polypeptides have this structure where you've added an amino acid from the amino end onto the carboxylic acid end here, starting over here and reacting with that. And we've regenerated, remember I said I was going to cheat a little bit and protonate that carboxylic acid. Well, look at here, this is the C terminus of this dipeptide, and it has exactly the same structure as the C terminus, if you will, of this amino acid. And that means we can take another amino acid and do this whole reaction again and create a tripeptide from this dipeptide. Okay, and from the tripeptide we regenerate this carboxylic acid end, another amino acid come in, and we can make a tetrapeptide, pentapeptide, etc., etc. And you can build up these polypeptide chains into literally thousands and thousands of residues. They're all condensation reactions. We're removing water from these things. We can chop them back up with hydrolysis reactions catalyzed by proteases. So a couple things to remember here. This is the nucleophilic attack of the amino group on the carboxylic acid group, and we condense out water and we make a peptide bond. The other thing to be very sort of cognizant about these peptide bonds is that carbonyl carbon and that imido nitrogen, that bond between them has a very, very strong double bond character, which causes that system there to remain flat and planar and not rotatable. The bonds neighboring it between the alpha carbon here and the nitrogen, that's rotatable, and between the alpha carbon of the alanine group and its carbonyl carbon, that's rotatable. But the actual peptide bond here, that one right here, because of that double bond character, is not rotatable. Okay, So that's the formation of peptide bonds, condensation reactions between the amino group of one amino acid and the carboxylic acid of a second amino acid. Thanks for your attention. I hope these things are useful. Let me know if there's something you need that I can do to improve these things. Thanks again.